So welcome everyone also from my side. My name is Mathias Witte Paz and I'm a PhD student at the University of Tübingen in Germany. And today I will be presenting one project that we've been working with Simon Heilbronner's lab from the LMU in Munich, where we've been analyzing the uptake potential of siderophores in Corynebacteria. But first, let me introduce to you what are siderophores. And many of you might already know that iron is essential for different organisms. In the case of bacteria, for proliferation. However, in different environments, iron is only present in a really limited way, in a toxic way. So other organisms, or organisms in general, have developed a mechanism of siderophores to uptake iron from the environment. These are secondary metabolites that are going to be exported from the cell to bind iron and then be reuptaken by the organism itself to profit from the iron from the environment. As any secondary metabolites, siderophores are synthesized in biosynthetic gene clusters. So we're going to find different genes close to each other that are responsible for the synthesis of siderophores, and also other genes that are responsible for the transport of the siderophore iron complex. And here I would like to highlight one specific type of proteins, which are lipoproteins. These are going to bind the siderophore iron complex before actually starting the process of the uptake itself. And also I would like to shortly highlight that these groups of genes are often regulated by one specific transcription factor. So we are going to find one binding site upstream of the group of genes rather than upstream of each single individual gene. Since we're in the nasal environment, one of the most known organisms here is Staphylococcus aureus. And Staphylococcus aureus is known to produce two different kinds of siderophores. In this case, staphylophorin A and B. And the lipoproteins responsible for the uptake of SFA and SFB are HCSA and CERA. However, it is also known that Staphylococcus aureus is able to take siderophores from other organisms without actually being responsible or actually synthesizing them themselves. And this is the mechanisms by FUD1 and 2 and SSTD. So this means that siderophores are actually common goods in a community. This means that can be stolen by any other organism within this community, and even from other organisms that are not really able to produce any kind of siderophores themselves. And this was shown by our collaborators that Corynebacteria are also able to cheat on Staphylococcus aureus. They did this by doing some co-cultural with Staphylococcus aureus, and they were able to show that some of them are able to steal SFA, SFB, or both of them even. And they identified two specific lipoproteins that were responsible for the uptake, in this case, one for the uptake of both SFA and SFB, and another one that, at least for these two siderophores, did not show any phenotype. So the main question from our project here is, how are siderophores actually shaping bacterial communities? But we realized that we first need to answer one specific question, is which mechanisms are Corynebacteria using for cheating on Staphylococcus aureus and uptaking the siderophores that are being produced? In order to answer this question, we have been analyzing a data set provided by our collaborators that consists of 50 different sequenced nasal isolates from the Corynebacteria species that are spanning 6 different species with the gene distribution as seen here. What we also got was metadata from them if this species should be able to steal either SFA, SFB, or both of them, as you can see here. So our question was, are we able to predict, based only on the genomic sequence, if a specific organism is going to cheat on Staphylococcus aureus in this case? And we're focusing now first on the lipoprotein homology, since we expect these lipoproteins to have a conserved structure to bind the siderophore in order to steal it from the environment. Our first approach was to try sequence homology. However, the results were not showing actually, or they were not reflecting the metadata that we got from the wet lab experiments. Hence, we wanted to increase the sensitivity of our approach and actually include protein structure homology for our approach here. And for this, we've developed one pipeline based on Nextflow that is going to combine sequence and structural characteristics. And it actually has a similar structure presented before by Rob Finn in the morning keynote. In this case, we start here first by predicting all of the lipoproteins of our species, in this case the Corynebacteria, using signal P. Afterwards, since we're going to have many isolates that are similar to each other, we want to reduce the redundancy of a protein set by MN62 to get just one single representative for each cluster. We're then predicting the tertiary structure via collab fold. Why? Because we already have references from those lipoproteins responsible for the SID uptake of Staphylococcus aureus, and these are the 5 proteins I've introduced at the beginning of the talk. And by comparing these predicted structures from us and then the reference structures by a fold-seq, we're going to get a set of putative homologs of similar lipoproteins that we're then going to decluster. So instead of just taking the representative, we want to know for each single sample that we have, which are the putative homologs that are in the organism. Before applying this actually to our Corynebacteria set, we wanted to see if our approach was actually valid and we have correct results. So we applied this method to a well-annotated organism, in this case Bacillus subtilis, and we got 7 different putative homologs as seen here. And when we went to the literature, we were able to compare this with the SubTWiki, and we were able to verify that all of these 7 homologs are actually already known to be related for siderophore uptake. Moreover, 2 of these homologs, or 2 of these lipoproteins were already identified as homolog HTSA, so the SFA uptaking lipoprotein. So we then were confident and applied this to our Corynebacteria and got 354 homologs distributed across all of the species that we had in our data set. But we want to go a little bit further, and since we already got RNA-seq data for 2 of the species that we were analyzing in 2 different environments, one control environment and one iron-deprived environment, and we already got here a lot of differentially expressed genes. But we were still asking ourselves, where are the homologs that we identified? And as you can see here in this volcano plot, you're able to see that up in the upper right corner, we have the identified homologs that were returned by our pipeline. So we were confident, again, that the results that we got from our analysis are really related to our high iron homeostasis genes. However, one of the big questions that we still have not tackled yet is that which lipoprotein is actually responsible for the uptake of which siderophore. And sadly, I still don't have any conclusive results on this part of the specificity of the homology. So if you might want to talk a little bit about this, you can approach me at any break. What we also got, but still we have a huge set of homologs that are really interesting for our iron homeostasis. And we were asking ourselves, how are these genes being regulated in Corynebacteria? And as we've mentioned before, these genes are often regulated by specific transcription factors, so we expect to find only one binding site upstream of a group of genes. So what we did here was to expand our pipeline to also predict operons in our bacteria, and then by taking or identifying which homologs have we find, we are actually taking the putative promoter region, so the upstream region of the operon itself, so the first gene of the operon, such as seen in this part here, or if the homolog is not found in any operon structure, we're just taking the putative structure of the putative promoter region of this gene. Since we have quite similar data or similar samples, we're going to remove duplicates to avoid bias towards one specific species, and then we're providing this to Extreme to get a de novo enriched motif analysis and also a comparison to different databases of known binding sites. From these results, we're then focusing only on transcription factors that are really known to regulate iron uptake. And we're not only doing this for Corynebacteria, but also for Staphylococcus aureus, since we already know how Staphylococcus aureus is being regulated for our iron homeostasis. In the case of Staphylococcus aureus, we expect or we know that FER, the ferric uptake regulator, is regulating this iron homeostasis, as the name says. But for Corynebacteria, we actually identified a binding site for IDER in the upstream region of all of those putative homologs that we identified. So as you can see here, in all Corynebacteria, we find more binding sites for IDER and just a few of FUR. And only in Staphylococcus aureus, we found more FUR binding sites for the different homologs than IDER. When we went to the literature, we were able to confirm that IDER is already known to regulate iron in Mycobacterium tuberculosis, but also in Corynebacterium diphtheria. So again, this confirmed the results that we already produced. And when we want to do next is to actually consolidate the motif for binding sites for IDER for Corynebacteria, to then use this to scan the genome and see which genes are found downstream of our specific genes, and see if there might be also iron-related genes that we might overlooked by our approach. And with this, I would like to conclude my talk, where I shortly presented this cross-genus homology analysis for lipoprotein homology. And this is going to be a base to see how the microbiome is going to be shaped via siderophores. We are also planning to expand our method or our approach to analyze more than just the nasal environment, but also soil and other microbiomes. But this will be in the future project. And with this, I would like to thank our funding providers and our collaboration, and also all members of my group. And I was asked for my supervisor to promote here one PhD position that is open for my lab. So if you know someone that might have interest in having a PhD position in Germany, this is a really great group. It has nothing to do with siderophores. It has more to do with zebrafish and visualization techniques. But if you have any questions, you can just ask me at any break. And with this, I'm happy to answer any questions. Thank you very much.
Thank you, Mathias. Any questions from the audience? I have one question if you don't have from the audience. Here, please, go ahead.
So sort of question straight in on it. So when you're trying to match the homologs to the potential siderophore binder, have you thought about using AlphaFold 3? Because you've got a small enough search space that it's possibly computationally feasible.
The problem with AlphaFold 3 is it's not allowing to actually, you mean with the metabolite, or?
Yes, you can
it's still restricted to a specific kind of metabolites. You cannot actually provide it. I've tried actually, and it did not have any good results. But yeah, I was also expecting a little bit more from that part, but it did not really turn good results.
And which tools did you use in the homology search?
For the homology search, FoldSeq. So we're comparing everything from the structural level. So we're using FoldSeq to compare all of the PDB files that we're predicting and those references that we got from the PDB.
Perfect. Thank you. Any other question from the audience here, please?
Yeah, thank you for the talk. How common is this type of interaction for iron siderophores in nature?
I'm not really sure. I think it's going to play a big role. Actually, it's one of the first things that Simon Halbronner, so the one that actually started this project, has been looking at from the nasal perspective. But we do see that siderophores are common in other environments, and this might, since they are common goods and they're really exported from the cell and available for everyone, so to say, this could shape many communities from the bacterial side. So I'm not really sure. We wanted to expand this further, but I do expect to find it in multiple environments.
So that means that so far this interaction is only
in the nose.
Yeah.
I know that siderophores are produced by many organisms. Even plants produce siderophores. So as long as siderophores play a role, this could be used by any kind of organisms of this close environment. But like I said, so far we have just focused on the nasal environment, but I would predict that it's also in other environments the case.
Thank you.